The topic is lactoperon. Lactoperon, the full form is lactose operon. This is also known as inducible operon, and it has been studied in detail in case of Escherichia coli. The work has been done by Jacob and Monod in 1961, for which they got the Nobel Prize in 1965. What is an operon? Operon is a small segment of DNA, which we can see it over here, and it comprises of structural genes and several other genes. So there are three structural genes shown over here: structural gene Z, Y and A. Upstream of the structural gene is O, which stands for operator gene, and upstream of operator gene is P, capital P, which is a promoter gene, and upstream of promoter gene is regulator gene, which is denoted here by I, capital I. This lactoperon is usually in a switched off mode. That means the E. coli is grown in a glucose medium and there is no lactose. So when there is no lactose, it is in the switched off mode. So how does it work? When there is no lactose, what happens is the regulator gene, it transcribes and then it produces protein which is known as accessory protein, and it is known as repressor, which is, I have shown it with a pink color. This is a repressor. What is the function of the repressor? That in absence of lactose, this repressor goes and binds to the operator. When it binds to the operator, what happens is the operator blocks the promoter. Promoter is the site where the RNA polymerase gets attached. It recognizes promoter and gets attached over here. So when the operator is blocked, then RNA polymerase cannot move. And since it cannot move, then these genes cannot be transcribed and neither it can be translated. So it remains in a switched off mode. Now when we look at the second part, this I am showing you as switched on mode. How does the operon get switched on? That means if the medium is supplemented with lactose, what happens is some of the lactose diffuses inside the cell. This is the cell membrane I am talking about. This is cell membrane which has been shown there also. This diffuses, and this diffusion is helped because of the enzyme known as permease, which is available in the cell in less quantity. So when the lactose diffuses in, it forms a structure which is known as allolactose. The lactose which is going in now becomes allolactose. Now this allolactose has a very high affinity of binding with the repressor. That means the repressor can now binds with the allolactose. When it binds to the allolactose, then this operator region is set free because of the repressor inducer complex which is formed. Here the inducer is lactose, alright, and the active form is allolactose. Once it is in allolactose form, then only the repressor is going to bind to it. So the structural changes takes place and hence the operator is set free. Once the operator is free, the RNA polymerase which is bound to the promoter region now can move ahead since there is no hindrance over here. So it rolls out, and then these Z gene, Y gene, A genes they transcribe and an mRNA is formed. This mRNA, with the help of ribosome and specific tRNAs, is translated into enzymes, and three enzymes are formed. Z forms beta galactosidase enzyme, Y gene forms permease enzyme, and A forms transacetylase enzyme. The beta galactosidase is the main enzyme which breaks up lactose into glucose and galactose. Permease we have already mentioned it helps in the permeability of the cell membrane. That means more permeable, the more of lactose will enter into it. Trans-acetylase function is not very much clear, but it says in some, they mentioned that it helps in the transfer of acetyl coenzyme into beta-galactosidase. And we call it as a switched on mode, and this will continue till the lactose is available. Once the lactose is converted to glucose completely, then again it goes back to the switched off mode. Because when the glucose is high in the medium, there is something called CAMP, cyclic AMP. Its concentration is low. So more the glucose, less is CAMP. But less the glucose and more of, that means if the glucose is completely utilized and then lactose goes into the medium, which is the inducer, then CAMP forms a complex with CRP. So CAMP, CRP complex is formed, which enhances the operon. So this is about lac operon, which has been studied in E. coli.